This part was points down.
Okay, let's try this again. I don't know if it's recording, but if it's not, I'll do something. So anyway, just so people know that because of there's a meeting to deal with like post-election stress and conversations and stuff that's going on from 2 to 4 that I want to attend, and I urge many of you to attend as well in Tucker. I'm going to be a little bit late to my normal 3:30 time at the Daily Grind, but I'll be there from 4 till 5. There is a review session tonight, as usual, in the teaching lab. So, and I also have a special additional one this week because I'm sensing, I'm getting feedback from the TAs that there's a little bit of angst amongst the students because you realize that this is the last week of the lab. And there's no labs on the week of Thanksgiving. But then when you come back, it will be the laboratory practical. So we will take as much time as necessary in tonight's review session, as well as Wednesday's review session, and next Monday's to go over materials to help you prepare for the lab practical. I'm also going to put together kind of a study guide, maybe even some problem sets on dilutions and things like that I'll get you on Blackboard. So if you're starting to really sweat that 50-point lab practical in the last week of classes, just take a deep breath. We're gonna get through it. It's gonna be fine. Okay, so what we want to do today, and I don't know why this is projecting with a green background. It probably is my computer, but now we want to move on to another RNA virus. So we talked at length about coronaviruses and specifically about poliovirus. So that was an RNA virus that was single-stranded, but its single-stranded RNA was of the plus sense. So in other words, it had RNA that could be translated and would be translated directly upon infection of the cell. So it behaved exactly as if it were messenger RNA. Now as we move on and talk about a general group of viruses known as the orthomyxoviruses, this is also an RNA virus. It's also a single-stranded RNA virus, but this virus is of the negative sense. So in other words, this messenger RNA, or this RNA, as it would be introduced into a cell, could not be translated, no way, no how, into proteins. So you'll see that in negative sense viruses like influenza, which is the example that we're going to use for orthomyxoviruses, that negative strand RNA genome must first be turned into a positive strand of RNA, and that gets used as messenger RNA. So this is just some general information on orthomyxoviruses and influenza in general. Really, we're just going to refer to it as influenza because for the most part, among orthomyxoviruses, you and I are mostly just concerned with influenza A or possibly influenza B. Influenza A is the type of influenza that gets most of the press. This is the type of influenza that has the ability to cause major pandemics throughout the world and has caused major pandemics throughout the world, one of which you even lived through, the swine flu. There is another form. No, I don't want to do that right now. My computer is really... there is another form called influenza B, which exists only in humans. Influenza A is kind of an interesting virus because it has the ability to infect birds and pigs as well as humans. Influenza B, on the other hand, doesn't cause major pandemics, but it does cause major epidemics in humans, and it only infects humans. And so when you get your flu booster, and you will all get your flu boosters, that will also have, within that cocktail, it will have an influenza B, one that's most likely circulating in the population now. But for the most part, we're going to talk about influenza A because it's just sexier. It has a lot more known about it. So it has a negative strand genome, and the genome is segmented. So it's not a single chromosome. It's in 8 different segments. And that's what's shown in the middle of this. So each one of these segments is meant to represent a single-stranded RNA encoding, for the most part, most of these encode a single protein. 2 of them encode 2 proteins. And what you're seeing here in the artist's attempt to represent this is that the RNA is actually coded and associated with viral proteins. So there are proteins called the nucleocapsid protein, shown here as these red dots covering this thing. The balls here on the end, on the left of these, are actually complexes of viral replicase. This is an important point. I keep I keep really hammering in on this because this is the enzyme that has the activity of RNA-dependent RNA polymerase. This virus, unlike the polio virus, needs to bring this activity with it preformed. One of the first things that the polio virus did was to translate its genome into a large protein that had proteolytic activity and had the ability to make its own viral replicase. Not the case with influenza virus because the genomes cannot be translated as they're put into the cell upon infection. So these viruses must bring with them, and they do, as shown by these little white balls on the end of the genomes, viral replicase, RNA-dependent RNA polymerase. So just in terms of anatomy of this virus, you have other viral proteins in here which are called the matrix proteins. And then outside of that matrix protein capsid lies a lipid bilayer. This lipid bilayer came from the previous cell that was infected, that produced this virus. But within that lipid bilayer, coming from the previous respiratory epithelial cell that produced this virus, there are at least 3 viral glycoproteins existing. There are occasionally some host proteins left behind, but by and large, these viral envelopes, this lipid bilayer, is pretty much exclusively studded with viral glycoproteins. The 2 most important ones, or I shouldn't say the most important ones, but the 2 most famous ones, the 2 that you've probably heard of in other classes, are the hemagglutinin, or the HA. This is an adhesin molecule. This is what's going to allow the virus to attach to another cell, either in that same host, or if the virus happens to get coughed out, it may infect another person. The other major glycoprotein that studs this outer lipid bilayer is the neuraminidase. We'll talk about this. The neuraminidase, you can tell from that name, is an enzyme ending with ASE. This is an enzyme that has the ability to cleave hemagglutinin away from its receptor, and we'll talk about the importance of that. And then perhaps a neglected membrane protein that studs this lipid bilayer is this M2 protein, or matrix 2 protein. So here M stands for matrix. There's matrix 1 protein, which is the inner core, the viral protein which lies just inside the lipid bilayer. And then this transmembrane protein, which actually gives access to the outside of the virus, a channel into the internal portion of the virus. This M2 protein is actually a proton channel, and you'll see the importance of that in a little while. These caps on here are actually made up of 3 different proteins which collaboratively make up this viral replicase. These are the PB1, PB2, and PA proteins. Collectively here, so you can remember the function of these proteins because P stands for polymerase. This is the molecule that does RNA-dependent RNA polymerase activity. This molecule collectively, when it makes an RNA copy of an RNA genome, has an error rate of about 1 every 10,000 base pairs, which is pretty not, I have a hard time with English language today, is not very good. So that is an error rate much higher than DNA-dependent DNA polymerases. And that's because RNA-dependent RNA polymerases do not have proofreading activities. DNA-dependent DNA polymerases do. So this means that virtually every virus that gets produced, on average, because there are about 10,000 nucleotides of RNA sequence combined between these 8 genomes, every virus produced has at least, on average, one mutation in it. This is why these viruses evolve so quickly. We'll talk about something called antigenic drift, how these viruses can mutate quite rapidly over time. That's why we need boosting, or one of the reasons why we need boosting to our vaccines, because these polymerase molecules, although they're very efficient at creating RNA from an RNA template, they are very error-prone as well. So these influenza viruses, so that's just a brief overview. I want to talk a little bit about the disease because that's what I find most, well, I don't know if I find it most interesting, but I do find it interesting. There have been three major pandemics, and the key word here is major because in reality we could call a fourth pandemic, the one that you lived through in 2009, the swine flu pandemic actually was determined by the World Health Organization to be a pandemic. It just didn't have quite as much of a mortality associated with it as the Spanish flu, which killed perhaps 20 to 50 million people. That estimation always blows my mind for a couple of reasons. First of all, that's a huge number of people, and it's also a huge range. You would think 1918 wasn't that long ago. I mean, my grandparents were alive during that. I knew people that lived through this. Yet, we still don't know whether 20 million or 50 million people died. In either case, that's a hell of a lot of people. We've never lost that many people from any of the subsequent influenza pandemics that have occurred, such as the Asian flu in the late 50s, and one that I lived through as a child, the Hong Kong flu. Each of these killed, a couple of million people, virtually nothing compared to the Spanish flu. But so these flus have had major effects on the human population and public health. So influenza virus is the etiologic agent of both epidemic and pandemic influenza. And the virus, or the flu itself, has been known, it was actually described by Hippocrates. So it's been around for a long, long time. Now, I'll talk a little bit, because I like history, talk a little bit about this 1918 Spanish flu influenza. So first of all, it has nothing to do with Spain. So here's just a historical nugget. This will never show up on an exam, but if anyone ever asks you why they call it the Spanish flu, it's because this flu actually started in 1917, and it was raising such hell with soldiers in World War I in Europe that the military censors did not want news to get back to the Allied countries that so many young men were dying of influenza. But Spain was neutral during World War I. So their press was not under the same censorship that the Allies were. So all of the people reading stories about this terrible influenza that was going on in Europe were always reading it in Spanish newspapers. And so they believed that Spain was the only place that was really having to deal, or at least bear the brunt of this influenza. And Spain really was not where this virus came from. But that's the genesis of that name. But if you look at the typical mortality patterns of influenza, these still hold today. If you look at this dashed line, so on the x-axis here are ages of people who died during a typical influenza epidemic, in groups going from babies through elderly people. And you can see that here with the dashed line, you can see that there are two peaks in the types of, or the ages of people who die. Either very young, under the age of 4, or generally the more elderly people. This still holds today. I mean, influenza is a major cause of death. This year's influenza, when it takes off, will kill thousands, if not tens of thousands of people in the US alone. Most of those people will either be small children or elderly people. But look at what happened in 1918. This spike here, where you had this tremendously much higher death rate among people in the primes of their lives. So people, just a little bit older than most of you, 25 to 34. These were the people that were dying, and this is why soldiers in World War I were experiencing such problems, is because this particular strain of flu was much more virulent for people of that age. People actually, in my age group, this always depresses me. When I first started lecturing, I was in this age group down here. Now all of a sudden I've moved up, I'm in the same age group as people in their 60s now. That's kind of scary. But we were actually much less likely to die of this same infection that was killing people in their 20s. That's pretty bizarre. People still can't really fully explain that. I think the best possible hypothesis is that the people who were in their 50s and 60s had perhaps experienced a related influenza sometime before these young men and women were born. So sometime in the 1800s, perhaps something related to the Spanish flu, but not as severe, swept through the population, so that populations in people my age had some degree of immunity, so the disease wasn't as severe. But in either case, this was a very frightening thing for people. People dying in the primes of their lives. So I mentioned, going back now to the molecular biology of influenza virus, that this is a segmented genome. So here are the 8 different genome sequences. You do not need to memorize what each one of these codes for. It'll become obvious what it is that I want you to know. But one of the things that I put this up for is that you'll see that for each one of these genome segments, there is typically one protein produced. So either of the 3 proteins involved in viral replicase, PB1, PA, PB2, the hemagglutinin, the nucleocapsid protein, the neuraminidase. But there are actually 10 proteins that are produced by an influenza A virus, despite the fact that it has only 8 genome fragments. So these last 2 genome fragments, 7 and 8, are expressed in 2 different forms, alternatively spliced forms. So this is a little bit unusual. Those of you who are taking cell biology or have taken cell biology will find out that our cells actually put a great premium on making certain that we don't export unspliced messenger RNA from the nucleus of our cells into the cytoplasm to be translated. So influenza virus, and the next virus that we'll talk about as well, retrovirus, HIV, have come up with ways to circumvent those safety nets that our cells have to prevent unspliced forms of RNA from being transported into the cytoplasm and translated. So that's what's happening here. For instance, in segment 7, which encodes the 2 matrix proteins, in the unspliced form, it produces that larger protein, the M1 matrix protein. That's the one that lies just inside the envelope of the virus. But at some frequency, messenger RNA made from segment 7 will be spliced, and an intron will be removed. And so what gets transported out is a different form of the messenger RNA, which creates a completely different protein from the same RNA. So this M2 protein, this is the ion channel, the proton channel, that you'll see why is important. And the same thing with segment 8. In the unspliced form, it has the ability to produce NS1, which stands for non-structural protein 1. This protein is still, or the non-structural proteins are still a little bit mysterious in what they do. It can also be alternatively spliced, so an intron removed and a different form of the messenger RNA will be transported into the cytoplasm to be translated into non-structural protein 1. So 10 proteins, 8 segments, really kind of simple in a way, but a little complex at the same time with this alternative splicing. So we won't talk any more about influenza B or influenza C because influenza C is very rarely a pathogen. Very little is known about it. Influenza B only infects humans and it doesn't cause pandemics. So we won't waste our time on it. This is just a picture that I stole. I don't even know where I stole this from. I like colors. So this is just another way to look at the influenza virus. And the only part that I'm really interested in you looking at is, so here is, shown here in big relief, the lipid bilayer, the envelope of this orthomyxovirus, this influenza virus. And you can see 2 major species, if you will, of proteins. The hemagglutinin protein shown in blue, the neuraminidase proteins shown in red, and the color is not coming out here very well. There's actually a third, but much less frequently found protein, this ion channel, the M2 protein. So this numerically, if you looked at all of the proteins exposed on the surface, would be probably the least that you would find in terms of numbers. It's mostly the hemagglutinin protein and the neuraminidase. So let's first talk about the hemagglutinin protein. So that's the protein, as I mentioned, that's an adhesin. This is the protein that initiates the virulence. This initiates the pathogenesis of influenza. So as your respiratory tract gets exposed to somebody who very rudely coughs in your direction, or if you touch something that somebody has coughed on recently or sneezed on or touched with their own hands and you touch your own face. This is why hand washing is so important during flu season. Because you think about the number of times, if you're somebody with the flu, you rub your nose, well, even thinking just to scratch your nose, or and then you touch a telephone, or telephone's not the same thing anymore. Everyone has their own telephone. Telephones used to, back in my day when everyone shared a phone in the house, that was a major fomite. That was how flu got spread. You guys all have your own phones. But think of things that you touch in common, like doorknobs, water fountains, things like that. And then the next person touches it, and without thinking, they rub their eye. That's why hand washing is really important. Because the initiation of this virulence has to do with the hemagglutinin protein binding to a glycoprotein on the surface of your respiratory epithelial cells, somewhere in your mouth, in your nose, in your throat. Hopefully not in your lungs. I'll talk a little bit about this later. One of the reasons why the influenza virus of 1918, we believe, was so much more virulent was that its receptors were found much deeper in the lungs. And that's not a normal place for influenza to replicate. So the more virulent forms of influenza tend to replicate deeper in the respiratory tract. Typical influenza virus that most of you have been infected with, this sialic acid glycoprotein, or this glycoprotein with the carbohydrate sialic acid, is found predominantly in the nasal cavity and in your upper respiratory tract, perhaps as far as your bronchioles. That's where, those are the cells that become infected. So what's shown here, each one of these little balls is meant to represent an amino acid, I believe, or perhaps some sugars. But at the end of this, there needs to be a sugar called sialic acid. So don't be thrown by that name. Sialic acid is a carbohydrate. It's a sugar. There is an older name for it, and you need to know both names because it will make things make a lot more sense to you. The old name for sialic acid was neuraminic acid. If that name sounds familiar, that's because we haven't yet talked about it, but I've mentioned the neuraminidase, this other glycoprotein. So sialic acid or neuraminidase acid, when it's in the conformation, thank you. I always need somebody to feed me words. If it's in the right conformation and the right binding, and at the end of this glycoprotein, the hemagglutinin will bind to that to initiate the infection. Now, here's just another way to look at it. So for the biochemical oriented people in here, although this isn't projecting very well, this is the chemical structure of neuraminic acid or sialic acid. This needs to be the terminal sugar and in a particular linkage at the end of a glycoprotein in order for the hemagglutinin to bind. So the hemagglutinin protein tends to exist in this lipid bilayer, the viral envelope, as a trimer. So 3 identical protein molecules of the hemagglutinin inserted in this lipid bilayer. And here you see that M1 protein, this matrix protein that lies just below that. Here's the other protein, the other major glycoprotein that the virus puts into this called neuraminidase. So the first step in the pathogenesis of any infectious disease, for the most part, I can't think of an example that it wouldn't be offhand, is attachment to a naive host. So that's accomplished by the hemagglutinin binding to sialic acid on the surface of respiratory cells. But as it turns out, sialic acid is pretty widespread as a member of the glycoproteins on all of our respiratory tracts, but not so much elsewhere in the body, with the exception of red blood cells. This is where hemagglutinin gets its name from. It agglutinates red blood cells, so hemagglutinin comes from that. As it turns out, birds, so the other animals, especially aquatic birds, tend to have sialic acid receptors, not so much in their respiratory tract, but in their gastrointestinal tract. So flu in birds is predominantly a gastrointestinal disease, as opposed to in humans, where it's more of a respiratory disease. And that really has to do with where these sialic acid containing receptors are found. In birds, they just happen to be in the gastrointestinal tract rather than in the respiratory tract. So the hemagglutinin will bind to that sialic acid containing glycoprotein, and that will initiate endocytosis in a permissive cell. So note the term permissive, because I'm going to show you how influenza binds to red blood cells. But red blood cells cannot undergo endocytosis. They don't have the ability to take things into vacuoles, whereas our respiratory cells do. The cells that line our trachea, the cells that line our nasal mucosa are all capable, when influenza binds, to take those cells into vacuoles intracellularly by receptor-mediated endocytosis. So typical clathrin-coated pits form, and the virus is taken up. Now, during this initial stage of infection, neuraminidase can play a small role, because if the virus happens to bind to a non-permissive cell, it could be a red blood cell, but red blood cells shouldn't normally be exposed on the surface of your respiratory tract. So if that were the case, that would be a very abnormal situation. But let's use that as an example. If an influenza viral particle attached to a red blood cell rather than a respiratory epithelial cell, after a few minutes, the virus would actually be able to release itself from that non-permissive cell because it hasn't been taken up. The neuraminidase enzyme has the ability to cleave this sialic acid off of the glycoprotein that's bound to this hemagglutinin, and thereby freeing the virus from that non-permissive cell. But its role at the beginning of the infection is probably very minor. Neuraminidase has probably its major role at the end of the pathogenesis, or at the end of a life cycle of influenza virus. As influenza viruses bud off, newly synthesized, newly created from a respiratory cell, they have hemagglutinin all over themselves, and they can get literally tangled up and bound to the same cell, or even in this, neighboring cells that are perhaps infected, and stuck there. And so these would not be viruses that would be easily coughed or sneezed out to spread to new hosts. So the neuraminidase, in terms of spreading the virus, becomes critically important, because now it will free that virus by cleaving sialic acid off from this hemagglutinin, allowing the virus to either infect another cell within that same host that perhaps hasn't been infected, but I think more importantly, it allows that virus the freedom, if you will, to potentially infect another host. So hemagglutination. So I did my doctoral work on characterizing hemagglutination, so I like this stuff. So I did hundreds and hundreds of these assays. But remember that red blood cells are not a natural host for influenza. But the way people can measure whether the virus is present is oftentimes by measuring for antibodies against this hemagglutinin. But if you take a red blood cell, that's what RBC stands for here, that has sialic acid containing glycoproteins on it, the influenza virus will bind to that. It's not going to do anything to that cell. It can't get inside that cell. And if it does get inside that cell, our red blood cells don't have a nucleus. And as you're going to see, this RNA virus requires a nucleus for its life cycle. So the red blood cells are a complete dead end for this virus. But what you can do is take these viruses, add them to red blood cells, and put them in these round bottom tubes. If you just take red blood cells with no virus and put them into solution into this tube and wait a few minutes, the red blood cells will just slowly roll to the bottom, and they'll form this nice tight little button at the bottom of the tube. That's no hemagglutination. That would be there are no viruses present. However, if the viruses are present, they can form bridges between these erythrocytes, these red blood cells, so they can do what's known as hemagglutination. And so when you see that, if you add virus to that same solution of red blood cells, rather than have the viruses roll gently down into a pellet, they form this mesh work, this lattice-like network that doesn't form a button at the bottom. So you get this kind of a veil at the bottom. So that's what's shown here in real time. This is a dilution of viruses. So here on the right-hand area of rows, these are round bottom wells that have just had red blood cells added to them with no virus. And 15 or 20 minutes later, the red blood cells have just rolled to the bottom into buttons. What happened in this row here is that before the red blood cells were added, a lab worker did a serial two-fold dilution. So they added a potential viral preparation of influenza to this well, and then they took half of that volume and put it into this well. So diluted at 1 to 2, half into this one. So this is a 1 to 2 dilution, 1 to 4, 8, 16, 32, 64. So you've all done 1 to 10 dilutions, but you can do dilutions in any factor that you want to. The most common are 1 to 10 dilutions, as you've done in the lab, but also 1 to 2 dilutions are very common for diluting antibodies or for diluting viruses. And so you can see that the virus becomes more and more diluted, but there's still sufficient virus until you get to at least here and maybe here to hemagglutinin, to bind to those red blood cells to prevent them from forming a button. But eventually you dilute beyond the point where there's sufficient number of viral particles to produce that hemagglutination, so they're beginning to form buttons again. The more realistic or the better use for this is a serological or antibody test, where you would actually add antiserum, potential antiserum from you or I, to see have we had been infected with this virus. Because if we produced antibody against the hemagglutinin, our antibodies could actually inhibit this hemagglutination. So we can actually quantify the amount of antibodies present as a means to see how strong our immune system is and how recently we were infected with influenza. So these hemagglutination tests, even though this is not a permissive virus, are important to understand because they're important diagnostic tests. And since I stole this from a French website, you have to understand it in French. So this hemagglutinin protein allows for the virus to bind to now permissive cells. Forget about red blood cells anymore. We're done talking about erythrocytes. So once they attach and receptor-mediated endocytosis occurs, what's happened really is that the virus, the cell, it's really a Trojan horse. Think of it this way. The cell is intending to be able to destroy this particle. So this virus that's being brought in, it could be a bacterium as far as the cell is concerned. But the material that's within this endosome, the cell wants to kill. It wants to destroy it. This is a self-defense and an innate immune system. So the endosome function that's formed here after receptor-mediated endocytosis is to break down the internalized contents. And one of the chief ways that occurs is by acidification. So there are proteins that line the membranes of these endosomes, which will now pump protons inside that inside that endosome, in order to kill the bacterium or the virus or the parasite or whatever it is. And in many cases, this is functional. But in this case, acidifying the endosome actually falls into the plan of influenza. Influenza has actually evolved to need that acidification. So that acidification that's going in, the protons being pumped in there, are going to change. So as pH drops, the protein conformation of that hemagglutinin protein changes. It just switches completely from one form to another. And that massive change in the conformation of the hemagglutinin will actually bring the lipid bilayer of this virus so close to the lipid bilayer of the endosome that they literally fuse. Think in your mind of playing with soap bubbles when you're a kid. You can take two soap bubbles and put them together, and for a few seconds, one soap bubble sits on top of another. And then all of a sudden, one soap bubble will just be subsumed by the other, and that bubble gets a little bit bigger. That's very analogous to what's happening here, is that by changing the conformation of the hemagglutinin, only when acidity is involved, tells this virus that it's now inside an endosome. And so now what it needs to do is to change that hemagglutinin conformation so much that it actually pulls those two lipid bilayers so close together that they actually come in contact with one another and fuse. And when that happens, what's dumped out into the cytoplasm of the cell are these viral ribonucleoproteins. That's what VRNP stands for here. This is that complex of all of the 8 segments of the influenza virus coated with nucleoprotein as well as the, all of the subunits of the polymerase. And those are actually going to be trafficked through a nuclear pore complex and into the nucleus. That acidification that occurs in this is actually, well, the acidification is driven by the host. But there's a viral protein that actually helps to get those protons inside the virus so that the hemagglutinin protein can change its conformation. That's the function of that ion channel that I talked about, that proton channel, M2. M2, or matrix 2 protein, is actually a pore which will allow only protons to go through it. So now as protons get into that endosome, the M2 protein allows them to get access inside of the virus. So now the hemagglutinin is exposed both inside the virus and outside the virus to low pH. So that changes the conformation of that protein. So that's what's being shown here. So here you have an influenza virus binding by its hemagglutinin protein to a sialic acid containing protein. And now receptor-mediated endocytosis has occurred. And so you see in this first blow-up here on the right, I know this doesn't project very well, but hopefully it looks better in your PowerPoint. You can see represented here at the bottom with these things that look like golf tees in them. This is the endosome membrane. And these things that look a bit like golf tees, actually with golf balls on top of them, are actually the sialic acid containing receptors. And these, what look like golf balls attached to them, that's a portion of the hemagglutinin protein. So in this particular conformation, this is the conformation that those 3 hemagglutinin molecules, remember the hemagglutinin comes in a homotrimer, so 3 identical proteins clustered together. And so you can see them here, 1, 2, 3, with a section bound to that receptor protein. But this is under neutral conditions. So this is what happens immediately upon attachment to the cell, and then the cell is endocytosed. But now, if you move a little bit down here in this pathogenesis chart, you'll see that the host begins to pump in protons to lower the pH into that endosome. And some of those protons are going to not only acidify the contents of the endosome, they're going to get access inside the virus by means of that matrix 2 protein. So now the hemagglutinin protein, exposed to acidity both inside the virus and the portion of the protein that's exposed within the endosome, will now change conformation. So you notice here that these regions that had been bound to the sialic acid receptor have changed their conformation. And then if you come down a little further, here's the final conformation that the hemagglutinin protein takes on under acid conditions. And so you can see how much closer the lipid bilayer of the viral envelope is to the lipid bilayer of the endosome. These become so close that that really greatly increases the efficiency or the frequency with which those 2 lipid bilayers will fuse. And when that occurs, that's what's shown here between step 4 and 5, is basically the viral envelope turns inside out. And all of the materials inside of that virus are now spilled into the cytoplasm. Now, all of those viral ribonucleoprotein complexes can find their way into the nucleus to begin the process of gene expression so that new viruses can be created. So here's just another way to look at it, and also to look at it in contrast to the next virus. We're not there yet, but when the next virus that we're going to talk about is a retrovirus. And so I like this slide because it shows the 2 different ways that membrane fusion can occur. Retroviruses, shown here on the left, like HIV-1 and influenza, are both enveloped viruses, so they have that lipid bilayer. We've already talked about here on the right how receptor-mediated endocytosis takes this enveloped virus inside an endosome, which becomes acidified, which will change the conformation of the hemagglutinin protein, which is going to cause a fusion between the viral envelope and the endosome, releasing the contents into the cytoplasm. Similar, but by a different mechanism, HIV, which also has a lipid bilayer from its previous host, has the ability via its receptors that we'll talk about in the next series of lectures, to bring the retroviral lipid bilayer or envelope so close to the cytoplasmic membrane of the infected cell, perhaps a macrophage, perhaps a lymphocyte, that it will actually fuse. And again, the internal components of that retrovirus are now dumped into the cytoplasm, where reverse transcription can occur and material ends up in the nucleus. So again, envelope viruses very frequently have mechanisms, and one of the things I want you to know is to be able to compare and contrast the way influenza virus mediates membrane fusion as opposed to retroviruses mediating fusion. You can't do that comparison yet. We haven't got to retrovirus. But at least now you know the first half of that exam question on how is it that the influenza virus mediate fusion of their envelope and the endosome vesicle. Now, in terms of chemotherapeutics, one of the reasons why it's important to understand influenza is that somebody figured out that you could actually block the ability of protons to get inside the virus. So you don't block the actual acidification of the endosome. That still occurs, but the drug amantadine, which is used quite frequently to treat people who have just recently been exposed or are just beginning to get sick from influenza, will limit the amount of infection that can occur, because what amantadine does is it's basically like a cork that fits into that M2 channel to prevent protons from getting in. So the acidification doesn't complete itself. It doesn't completely cause the conformation change in hemagglutinin that's necessary to fuse the viral envelope and the endosome membrane. So the influenza virus is stuck inside the endosome where it can't do anything. If this virus does not get access to the host nucleus, it's a dead end. So amantadine is very good at preventing that fusion. Now, there's another drug that you've probably heard of, and maybe even some of you have taken this if you've been exposed to influenza, and that's a neuraminidase inhibitor, oseltamivir, or Tamiflu is the more common name for this. Not Theraflu, that over-the-counter stuff that you get for sniffles. This is Tamiflu. This is actually another drug that can be used to inhibit other cells from being infected. So again, this is a drug that you give to somebody early on during infection to limit the amount of infection. Because what happens with neuraminidase is that as the viruses begin to bud from this cell, newly synthesized, that virus needs to be released from the cell. And neuraminidase is the enzyme that cleaves the sialic acid off of this receptor, which is holding on to the hemagglutinin. Tamiflu actually inhibits neuraminidase. So this virus ends up stuck on the cell that it just infected. So this person is much less likely to spread that infection to other cells in their respiratory tract. So it tends to limit the infection, hopefully just long enough for the immune system to take care of that virus infection. And it also hopefully helps to limit the spread of the virus to other people who are as yet uninfected. So that's why it's important that you understand these various aspects of influenza. Okay, everyone's favorite, I keep coming back to this, the 7-methylguanosine cap. So remember that in order for messenger RNA in your cells and my cells to be translated by ribosomes, got to have this 7-methylguanosine cap. So here's the 5' end of a messenger RNA made by your cells or my cells in the nucleus, transported into the cytoplasm. It's got this 7-methylguanosine cap on it. The ribosome recognizes and says, okay, let's translate this into protein. So any viral messenger RNA, in influenza viral messenger RNA that's going to be translated needs to have this 5' methylguanosine cap. Again, let's go back, so just so you know that I love compare and contrast questions. When we talked about poliovirus, remember that the poliovirus has evolved a way to have its RNA translated without having the 7-methylguanosine cap. Remember that picornaviruses like poliovirus have this trick that they do called an internal ribosome entry site, or IRIS site, a secondary structure that causes ribosomes to bind even if that RNA doesn't have a 5' methylguanosine cap. Influenza virus doesn't have that trick. It has a different trick. It's actually kind of cooler. What happens is that one of the proteins that comes in preformed, one of the subunits of that viral replicase, PB2, and again, remember, when you see this P, that means polymerase. This is one of the subunits of viral replicase. This protein, this enzyme, actually has the ability to do something called cap snatching or decapitation. This enzyme actually has the ability to take host messenger RNA in the nucleus and cut the head off it. It can cut this end, the 5' end, off of any and probably every messenger RNA synthesized in the nucleus of that cell. That is a great strategy from the virus's point of view because those RNA can never be translated by the host because they've lost their ability to be recognized by host ribosomes. So that's great. Viruses want to stop host translation because they don't have to compete with the host machinery to express their own genes. And it prevents the host from mounting an immune response or possibly even apoptosis. So cutting those or decapitating these messenger RNA is a wonderful thing if it did nothing else. But it does one other thing. Decapitation not only halts that host protein, but it now uses that head that it just cut off of the messenger RNA as a primer. So now it's going to use those caps that it's just snatched to prime the synthesis of influenza messenger RNA. So now the influenza messenger RNA that gets transported out into the cytoplasm looks just like host RNA. It's got a 5' methylguanosine cap. It also will have a 3' poly A tail. Ribosomes don't know that that's virus RNA. They lock and load and they translate these proteins, which eventually are going to kill that cell. So this cap snatching is really a clever strategy that the viruses have come up with. And so just before we leave, just make sure, just so as you think about studying for this exam, I always like to emphasize that for the short answer questions, I love compare and contrast questions. So think about, as you prepare for the exam, this process in orthomyxoviruses like influenza, and compare that to how poliovirus does the same thing. How does poliovirus stop translation? How does poliovirus make certain that its messenger RNAs are translated? And compare that to a completely different system in influenza, evolved completely independently, yet they do the exact same thing for these viruses. And we'll pick up from here on Wednesday. And I'll see you tonight at the review session.